hotel to
Grand Central Station.
Grand Central Station and back. Alright, so I'm here to talk to you about logistical issues. And I try to think about this in terms of the different types of trials that you all have done. Because some of you have multi-center trials, some of you have single-center trials, some of you are more developed in your thinking process, at least on what I saw on the synopses. I know a lot of what's going on this week. So I'm going to give you some tips about planning for logistical issues and to operationalize your clinical trials. And then I'm going to use two examples who it's not a surprise to Will Everett and also to Mojo and Domain that your projects were selected to just talk about some of the logistical issues that will come up. And I know a lot of this is in flux because of figuring out a lot of this. So my first slide is, so you want to do a clinical trial. Bill Clark was the director of the NINDS-CTMC at the University of Iowa where I worked when I started there. And he gave a keynote lecture, a presidential lecture at the University of Iowa. That was the title. So you want to do a clinical trial. Everyone should do one at least once. This is just a slide about, it depends on where you're coming from, on what the project looks like. There's all kinds of perceptions of reality, depending on who you are and how you're participating in the clinical trial enterprise. So the first thing about operationalizing a clinical trial is you need to begin thinking about the logistics early in the grant process. The goal is no surprises for the budget or during trial implementation. So how do you do that? First, you develop a good team. I've heard this talked about throughout the various small groups that I've been in, but first thing, who's going to manage and coordinate the project? It's not going to be successful if you are the only person who is involved as the PI. So you need to partner with a protocol coordinator, a project manager. There could be availability through your CTSA group. There could be, through your departmental funding. There's a variety of different ways that people do that. And then develop that person into a role if you really want to make a portfolio go forward so that they can assist you as you move ahead in your plan. You need to think about who's funding the project because that's going to have a lot to do with your budget drivers and some of the decisions that you need to make. So is it NIH funded? Is it industry? Or foundation money? Departmental funding? Who's going to help you design the project? Well, obviously the CTMC is going to help you with that. And a steering committee or a protocol steering committee can also get involved and get to the next level of planning for your project. And then recruiting and following participants. Are you going to have co-investigators? Are you going to have other clinical sites? Those are all logistics that need to get looked at. You need to pay a special eye to safety management, and Val gave a great talk on that this morning, all the considerations that go into safety management. Are you using a local IRB or will you have a single IRB? And is there medical monitoring going on, medical safety monitoring, independent medical monitoring, and DSME is the full-blown safety management program. So are you going to contract with vendors for drug distribution, for labeling, for placebo matching? How are you going to take a drug? Those are things you need to think about for central labs, for biologic samples, for central imaging. All of those are projects to work through as you go through. And being from a data coordinating center, we pay particular attention to how you're going to collect the data, how are you going to clean the data, how are you going to share the data at the end, because with NIH, you're required to share data. So are you going to use RedCap at your institution to have access to DCC, like we have in Iowa, and MUSC, and partner with them to develop the data collection and data aggregation. So to put this all together, you have to build the team, and the first thing is a well-written protocol, and I've heard a lot of this discussed in small groups. There's the grant application phase, and then there's the protocol phase. There are single-site projects where the local IRB will not require a protocol, but I highly recommend it as a way for you to formulate your thoughts, to think about exactly how this is presented, and identify what the pitfalls and barriers study materials for training and education, the data collection system, the data safety management, and SOPs, have them through your department for how you execute clinical trials, or through CTSA. There's a variety of places where those things are that you can reference, and then everyone's on the same page about that. Also, it's super important to have effective communication plans. Who's in charge and who's talking to who about what needs to happen and how that's going to happen. Of course, you need enough money to pull it off. So, don't design a project that is underfunded and you will have missing data, unhappy people. And that's always a struggle to make sure you have it. So I thought about this. I've given this lecture several times. We're trying to think about it a little bit different. But what are the 10 steps that I could identify that you need to go through, or the questions you need to answer to operationalize a clinical trial? First, you develop a protocol and an SOA. Do people know what an SOA is? Schedule of assessments. It's kind of a little grid. When I was a bedside research nurse, sometimes we didn't have time to read the whole protocol. All I would read was the SOA, and I knew exactly what we needed to do on that visit and what would go along. So that can also drive your payment system. So think about that as you're developing SOA. You're going to require that people have to do certain things for research purposes, and you need to figure out how to pay them for their time and or cover the cost of whatever that research assessment is or activity. Develop your budget, you define recruitment or retention strategies. It's super important to think about that from the beginning. You don't want to start out with bad assumptions that, and every site everywhere overestimates the amount of people you have available. So get as realistic as possible on who your available population is based on eligibility criteria and how they, and who might be amenable to enrolling in the project. Think about the central labs, think about central reviewing centers. That's especially important in almost every neurology trial that we've been involved in. There's always some kind of imaging that's involved. And if it's going to be used, especially as an endpoint, you want to make sure you're standardizing those readouts and getting the data available so that it's clean and actually usable in the format that the statisticians will need to do the analysis. Getting data collection, monitoring, and cleaning. Ongoing cleaning is something that probably gets overlooked unless you're working with the data coordinating center because there are just all kinds of pitfalls in Red Cap data. Lots of sites use Red Cap data, but there are issues there if you don't design the forms properly, if you don't ask the right questions at the right time, and if you're not looking for like triple logic and things like that that you need to clean up along the way. Think about your safety monitoring. Think about how you're going to train and retrain your site investigators. We brought that up yesterday. Just because you have a full-blown, session with them, the investigator meeting doesn't mean, a year later you might not want to do a check-in and make sure everybody's still following the protocol and or any of the assessment training that needs to happen. And again, how are you going to do data sharing and which will undoubtedly be required somewhere. So the first phases after you get your protocol ready, you got your budget going, is thinking about realistic timelines. The planning and startup phase involves site selection, just your site, or are there going to be few sites, many sites? I've seen all versions of that in projects that I've looked at through the course of this year. What do you need to do about drugs and supplies, devices? What do you need to do about training, the data collection system, the contracts and the IRB approvals? Hopefully you have someone in your institution help manage all of that. That's a real buzzword. And you need the right people to be working with that. Study execution, in addition to recruitment and retention and data collection, you need to develop some kind of tool so you can keep an eye on the project. If you have a single site, there are tools within Red Hat to try to dashboard to see how many participants you have and how many, where they're at in progress for data completeness. There's other things you might want to do for adverse events to keep an eye on that. But all kinds of reporting tools at whatever level your about is all about data cleaning and database locking. They're, certainly something you don't wait till the end. You need to be looking through your data set from the beginning and making sure that you have clean data points and are able to deliver that for analysis to the appropriate if you have a larger scale project, then you might want to look at on-site data monitoring. There's a lot of tools now that are used for remote data monitoring, also for risk-based monitoring, and working with statistical monitoring with different reports that you can do. And then often you need to receive and reconcile external data sets. So those need to be read into the overall study database, reconcile, which you think, well, of course they do this, they collect it. But it's amazing, things go awry between, especially if it's your primary endpoint, and the lab value. Do you know if they sent it off? Do you know if it was received at the lab? Do you know if you got the result back that you can tie to that? And what the time was for that results. And then, the final study report, however that's going to come together, and any kind of dissemination plan, which in this day and age includes reporting to patients, blinding people, the study PIs, the multi-sites, they're aware of the results, in addition to the manuscripts that you want to develop to disseminate what your findings were. And then keep track of your timelines so they can get drugged down on every point. So this is a tool that I wanted to make sure you were all aware of, the Clinical Research Toolkit that's on the NINDS website. You can get a lot of sample templates from the site. In particular, there's a protocol template. It's the next from the bottom, which it tells you what each section requires, and it tells you what the headers are and what is required to go through. Some things might not be applicable to your project, but it's a very good tool to use. We use these in Euronext. The PIs, the NINDS template. We've given them some suggestions for certain language that we know is going to be applicable across the network, so that can usually start on some of those things. And also, the other one I want to point out on here is the clinical trials checklist is very helpful, just for using notes, and also the data sheet. So one important question you want to ask early on in the process is this an IND or an IDE study? There are certain steps you need to go through to determine whether it is an IND or an IDE project. And different IRBs can help with that. You can write right to the FDA and ask them to weigh in on it. But it's important that you do that. And if you're not an IND, then you might want to get a letter from the FDA saying you don't require an IND for this. And then you want to do all that before you're submitting to the IRB. And NINDS generally requires that your IND is at least submitted to the FDA more than 31 days before your grant submission deadline, because they could kick back and that sort of thing. This is an important point about writing your specific aims, and what the purpose of that is. Again, this is one thing that we're in small groups. You have to have very clear specific aims so that, and often when we work on projects, we spend the most time on this, because the rest of it is but for a data coordinating center, that's like the operationalizing of science and things like that. But the specific aims for a data coordinating center to be very specific, and you want to make sure you're capturing whatever the RFA is asking you for, and then very clearly stating all that. And often your specific aims turn into your endpoints. It's important that you think that all through. You'll hear a lot, of course, that too many endpoints is not a good thing. One thing to think about is all primary and secondary endpoints have to be pre-specified in clinicaltrials.gov and you have to report on them. So that can be a lot of work. You have 25, 30 endpoints, secondary endpoints. And often we talk about making them exploratory because they're helping on how. So writing the protocol, the first thing you need to know is the research strategy and the protocol are not the same thing. research strategy is more about the scientific, and the protocol is how to operationalize that science. It's really helpful to include schematics, timelines, again, schedule assessments in the protocol so that people can actually see just a simple diagram. We're going to start recruitment. We're going to start on day 0. People are going to be enrolled. They're going to go into 1, 2, 3 arms, and they're going to be followed up this long. And any of that can include. Say that in words. Those are very useful items to include in your protocol. And I think it's also useful to develop an informed consent template because that's you trying to talk to an uninformed person in your field about what you're actually asking them to do and in addition to risk benefits, but just the procedures and all the different time points and when they'll be required. It helps you to get that straight in your thinking on what you are trying to accomplish. The budget, we've had a few talks about this, but the protocol and the scheduled assessments help define the scope of work and then what you have to budget for. Everything on that item, if you're saying that this is a research project, it's going to take someone's time or it's going to take money to pay someone to do those things. So you need to think through all that and where those things are, even if it's standard of care, because if it's standard of care, then you can't expect that it may come into your project because that might not be utilized the same way at a different site. So you can collect data from standard of care, but you can require you to tell people you're going to do that, and you have to can't double bill, do insurance, and do research payments to a site for doing what are standard of care assessments that have actually been billed to insurance. That's like a major red flag. You budget based on, you can do it either on unit activities or percent effort. So in Neuronext, we set up a patient or per participant, we speak subject fee, per participant fee, and it's basically takes the scheduled assessments and we talk about everything that has to happen at that visit. We think about how much time that's going to take. We think about if we have to pay to the radiology department or the study coordinator time to get that person from A to B. That can be a hidden cost because the pharmacy is actually a half a mile away, or and you have to get the drug there at a certain time. All of those things need to be thought about as you're designing a project. So what about recruitment and retention? We said start thinking about this early in the process. If you have a patient advocacy group that you're involved with, it's really useful to partner with them. They're very much open to partnering for any kind of conversation about participating in research. Things you need to think about for recruitment and retention. If you have recruitment materials, you have to, those are patient facing. You have to have IRB approval of those. You need to think about your posting on clinicaltrials.gov. You have to get that up and running within so many weeks or from first patient enroll. You have to make sure you have the clinicaltrials.gov posting up and ready for review by any sites. It's helpful to take pre-screening logs, especially if you're approaching people, they're difficult to find. I already asked them. I shouldn't ask them again next time they come around. And to think about, if it's multi-center, someone might want to be reviewing those in a blinded fashion to see what the barriers are that are prohibiting people from enrolling in the project. And that can result in changing your inclusion-exclusion criteria because there's a particular barrier they didn't see or didn't think would be a problem, but it happens to be. Remove those barriers, and then it's helpful to think about the site staff in the multi-center and what they're doing, and try to think about ways to recognize them for their work for recruitment and their work for retaining participants. In a network, we use different tools. I had a conversation with Will the other day about, can you send sites some kind of gift if they recruit the most, and ways that I've seen that be successful is anybody's eligible once they recruit a certain number, and the appreciation thing, but it's amazing how much that can help keep your sites engaged as they're going through all of the issues. So who's going to help enroll and who's going to follow participants if it's outside of just your center? This is not a good time to select your buddies. You want to have someone who has the appropriate clinical expertise, they have adequate staff to perform the duties, adequate facilities, there's a place to see people. Who was it saying? Study coordinator was in the bathroom. I think that was Tracy. On a study he was auditing several years ago, but she actually liked it because it was quiet. So we thought maybe flowers in the urinal would be an appropriate approach there. But you need to identify your eligible subjects, and for bigger multi-center trials, when you're partnering with investigators at sites, one important step is that there are no MBA members who are investigators. Drug supply is a big thing. A lot of these things I feel like I say, it doesn't just magically happen. And when you're in a big institution, you're used to, when I need a drug, I go to the pharmacy, I go to this paper in, or send in an order through Epic, and it appears. Well, the research gets a little more complicated than that. And we have seen, especially since the pandemic, the investigation drug pharmacies are really, I don't know what's going on. They're getting their budgets cut. They're getting more brazen about just refusing to do certain things, sometimes for good reason. We have several that will not allow on-site monitoring into the investigation drug pharmacy. So, and they won't use any systems we have. We need to use their system. And this is appropriate on several levels, but you need to recognize all of those things and figure out at your site level, what is it going to take to get your drug or your device. Different things come in. If it's a scheduled drug, another layer or something. Something has to say frozen at -70, another layer of something. If it's, do you need placebo matching? Who's going to do that? And placebo matching really means placebo matching. I've seen studies where people say, well, we'll just give them, this. It's supposed to look like that. Depending on what your project is, especially if you have subjective outcomes, which often we do in neurology, we need to match that a little bit better because the patient population can figure that out if they want to. If you're multi-center, then labeling and distributing the drug also becomes a big issue. There are several places throughout the United States that can help with this for academic centers. CMSU at University of Rochester worked with them at NINDS for quite some time, and this is their bread and butter. They do this, and they're much less expensive than some of the bigger industry partners. There's a VA system out of Arizona that also has a big pharmacy system. We've used the University of Cincinnati for a project that we had. They were willing to take this on several years ago. We're still doing that or not for multi-center, but some institutions will take it on, some will not. If you make that work their while. And all the accountability and shipping and temperature logs and all of those things, someone has to be on top of that. You need to pay someone to do those kinds of things because you're not going to be able to take care of that. Central labs, everybody's seen, lab kits at the site. All kinds of things go along with that, including did the tubes expire, who's getting the central lab kits out to the sites. Do they need to use a barcode scanner to read them in? Do the requisitions go where? Is there a laboratory manual? You need amazingly clear steps, especially the more sites you have, the more participants, to make sure that everyone is, if there are certain steps that are very important to processing up your samples. I will say here, that I've seen lab manuals where, purple top and green top, and this one they want you to spin at 35 degrees, and this one they want you to spin at 32 degrees, and instead of a range, just because that's what the lab was used to doing. So the more that gets standardized, the better off you're going to be with doing things in compliance. into, are you doing dump scans, are you doing phantom scans, who's collecting that data, who's QCing it, you have to read over to the MRI algorithm to correct for things. How do you ensure that every site is doing that? Chris talked earlier about the NF2 study, which it was just a tiny little, difference, but it was a difference detected, and that had a lot to do with how much rigor we put into the imaging, the standardization of the process, and when the decision we made. So here's my favorite topic: data collection. Make sure at the end of the day you're collecting your primary and secondary endpoint data. I have seen, especially in the flurry of everything, people getting too caught up in all like operational details or some other issues around, or secondary endpoint changed a little bit or whatever, and somebody missed it and we didn't respond to it, or I'm not saying we are, but I have seen this happen. So make sure when you're ready to launch, take a look and say, okay, we will map our CRF templates out and say, there's the primary endpoint, there's the secondary endpoint. Because a lot of it's operational stuff on top of that. Make sure you're collecting data in a useful way. Open text box are hated by almost everyone everywhere. If you're doing analysis, you might need that, and it also often needs PHI. So you want to make sure you're managing that in a way that makes sense. Don't collect data just because you might think that's interesting, because once you start doing all that, you get into subject burden and you get into the data management principles 101. If there's not an identified reason for that, will you share it at the end? That's another thing to think about. And anything collected with NIH dollars needs to be shared. There's something called the NINDS-CTMC data elements that you can actually download off the website, like CRFs. If you're not using them, because they're not available, or if you need to alter them a little, that's fine. But often, they're still doing this, but it used to be in our NOAAs that we had to use the CDs. This is like going back 12 to 13 years, very few. And if we weren't using them, we had to map. But that was part of the network building up the CD library. And then the data management. So how do you manage knowing if your data is accurate and clean? This is another thing that I think PIs and sometimes even the statisticians are like, oh, my data how do you assure that that this is the correct data and will lead to an appropriate analysis of your study? So at the very least, you have to enforce site self-audit. Every good site coordinator should be thinking about this stuff, but especially like in acute settings, someone can hand off a CRF or an EHR or something to a data manager on the project, and they are inputting something. This can apply to the data collection, this can apply to proper informed consent, this can apply to deviations, all of these things. Good sites will have steps built in where they are doing some self-auditing and not waiting until someone's going to come look at their records or ask for something and then find out that they're behind. Some sites just need to be shown certain ways to do that kind of thing, especially if it's a newer project. But it's going to be to your advantage hugely if you built in some kind of self-audit steps. If at the very least there's a weekly, okay, we enrolled this many people, how many CRFs do we have? How do we look at and see whether we have missing data or not? Much less how do we look at whether that data is correct? There are things for bigger trials with on-site monitoring, which I think most people are familiar with. Currently on-site monitoring, the old days they put Post-it notes on everything. But now there's often modules that people work in. But risk-based monitoring principles, I think, could be applied even to a single site project. If you're thinking about, what is the biggest risk? Well, first of all, I'm not getting my primary endpoint. But at the very least, you safety monitoring, NINDS does have guidelines on safety monitoring. It's quite extensive. It's been revised a few times, but I just pulled this one from last week when we put this together. And typically, NINDS thinks about this in 4 levels of safety monitoring. So it's your responsibility as the PI to make sure you're collecting the safety data. And then the lowest level is that the safety monitoring is done by the study PI and the local IR, which is perfectly fine. But you need to think about that and write that all down and then do that way. Next, you can have an independent medical monitor for single center intervention trials with lower risk. You have a study monitoring committee for single center intervention trials or moderate risk studies, and then some type of data safety monitoring board for multi-center intervention trials or high risk studies. At that point, you're going to want to be talking to your program officer about what the appropriate designation is and the development plan for all of that. So the trial master file, I won't go into a lot of detail about that, but because typically that is handled by some regulatory expert at your institution. But at the very least, make sure you're keeping track of IRB approvals and your enrollments, so that even if you're single site and local IRB review, so you have your IRB approvals and your informed consents for every enrolled participant. The level of requirements goes up depending on if you're multi-center, if it's an IND study of being regulated, at 1572s. What I always say here is, because I've seen people sign those and they have no idea what they signed. And it is an important commitment that you're making to the FDA that you will administer that project and you are ultimately responsible as the PI who signs on, including who you delegate to. So if you're not delegating to the appropriate people, it's not their fault, it's your fault when it comes down to it. Those are all the regulatory documents that are required to us. For multi-center trials, these are an example of the multitude of study materials that you might want to provide to the sites. Always an investigator brochure so that people understand what the current risks are in the product that's being evaluated. Manual procedures, lab manuals, pharmacy manuals, the templates, source docs that you're using now for direct data entry. We're seeing a lot more direct data entry, which creates other problems as far as how to verify. And so there are things that you can work with to make sure that that is the correct data. The training and startup, another thing, it's not just a fun time to get together with everybody. There are certain requirements for FDA regulated studies that training in GCP, everybody feels like they've had that 100 times, but you need to have documentation that you've done that, city training and all that, but especially going through the nuances of the protocol and the expectations of the investigators if you're multi-site. And that can be done through a variety of ways. We always do EC training, and things that are important are, especially if there's pharmacy or IWRS, the interactive web response system, how the randomization works, how the blinding works, and always you want to be thinking about that manuscript when you get out of the set your bar high. Think about all the things that you want to accomplish as you go along with the project. It's really helpful, especially if you have a multi-center trial, to publish a protocol paper so that you can reference that when you actually get to your primary results paper. You don't have to go over detailed protocol paper that you already published somewhere. A baseline paper can help, again, so you don't have to publish that or include that in your primary manuscript. But that requires a trial that goes on longer and you have baseline data to actually publish and stuff like that. And then there's all kinds of requirements to publish your primary paper anytime. The new policy for data sharing came out in January 23, and was revised in January 26. With the data sharing plan, the template, the first one was quite clunky and no one quite knew what it meant. And this revision that just came out in May is a little more user-friendly. But there are people at your institutions who can help you with that. We're going to submitted with your grant, and then the program officer's responsibility is to review that and accept it. The big thing is you have to name a repository where you're going to deposit it. The data sets need to be deposited at the time of the primary manuscript or at the end of the funding period, whichever is first. So you can't not deposit the data because you didn't get a manuscript out if you're out of funding. And NIH, I mean, everybody knows there's a lot of chaos over the last couple of years, but a lot of these policies have been around for a while, including CT.gov. You have to report your results within 1 year of the last patient primary endpoint. But NIH is starting to fine people for these, for violation of these regulations. So again, here's our 10 steps. We went through them already. And now I'm going to move into the 2 projects that we identified. And again, this isn't telling anyone out or do anything. It's just the things that I thought about when I read the synopses. And if you guys want to interject anything, feel free. But thinking about these things, protocol, SOA, budget, and the synopses, and I think everyone's going to hear these guys' projects once we break details about that. First one's Mo's, and this is endovascular outcomes in GLP-1 receptor for EBT, and 120 adults randomized at 2 sites within 1 institution. They're both at Pitt. I'm not quite sure how closely they're affiliated, but if you have 2 sites and they're not right together, that's something you have to think about as you operationalize. How do you make sure both sites are doing the same thing, if I understood this correctly? The study population in acute ischemic stroke due to a large vessel occlusion, planned for a thrombectomy and mechanical thrombectomy, but not receiving IV thrombectomy. And so those are things that I think about. First, that probably narrows your population some, because and again, I'm not an expert, just around it fairly often. So, does that narrow down your pool? Because it seems like IVT is the first pass for most people. And what does that do for the intervention of the drug if they have not had IVT? Don't think about not getting into the situation where you didn't know, or they, and I think this is because there could be some interaction between that product and the GLP-1 receptor agonist, so it's supposed to be correctly. So it's a safety issue. You want to make sure. So that needs to be well written out, and then and your sites need to be very aware of how they're going to know that. Sometimes that stuff's getting into the hospital, and then people are transferred over. Did we get all the information? Those are operational issues that have to get worked out as you're walking through. That's just enrollment, right? So the intervention is currently, I think, semaglutide, 0.5 milligram sub-Q prior to EDT, and then maybe on day 7, depending on this, that, and the other thing, which was written out in the concept. So it's open label, so it's not blinded. So then you have to think about your assessments and whether, if it's open label and well documented that who received this, then, and I think it's like, it's randomized, but, correct me if I'm wrong on this one, it's randomized, but it's open label on what the treatment is. So they either get the drug or they don't get the drug. So everyone pretty much. That helps with operational problems like blinding and labeling and distribution and all those kinds of things. But then you want to think about your endpoint assessments, and it doesn't matter who's doing those assessments and whether they know whether they got drug or not. There are a lot of endpoints in this one, and I think that's been talked about a little bit. I think that's also clinical endpoints and safety tools. So there is protocol-specific imaging, so now you have the training exercise that you need to think about. How are you going to make sure that everyone is adhering? Are they going to one imaging place or are there two different ones? No, there's two sites. If there are two different centers, then how are you going to make sure that everyone is adhering to that kind of training? How do you document all of that? And you can't just think, well, we told you to do it. I guess that's probably the biggest lesson I can give you. Just because you told them once or wrote it down in someplace, you have to have checks and balances in place to make sure, because it just dilutes your things when things happen that are out of range. There were multiple safety endpoints, but I don't know that you had time yet to think about your safety management plan, but that will need to be developed. Whether there's a medical safety monitor involved, the DSMP, how you're going to collect adverse events. We talked a little bit this morning about different projects where, I think Valerie was asked a question, if it's a single site adverse event, how do you figure out which aids to report? And one pitfall is thinking, well, of course that happened. They had this problem and that problem. So how do you define which ones were actually adverse events and then whether they were related or possibly related to the intervention? Outcome training on the MRS, that's we've had all kinds of discussions and models around all of that to make sure, and should they be a blinded educator? And then your data collection, EHR extraction, or data inputting into the Red Cap system. How is that going to happen so that you have your data plugged in? That's some of the things I thought about the most. Will's project was a phase 2A randomized double-blind placebo-controlled in individuals with ALS. So this is a larger scale trial in that it's 10 to 15 sites with 114 adults. And then you get into the problems of startup and training. You have more sites that needs more training and more startup issues on just getting them up and running and ready to go. So that all has to. And what I saw, this study was totally planned to go for over 24 months. So getting 114 people enrolled at 10 to 15 sites within 2 years of funding would probably be a big challenge. Unless you leverage the network and then and it's that quickly. The study population have to be diagnosed with ALS within 36 months of symptom onset. There's some things there that could be discussed because symptom onset in ALS, which symptoms are we talking about, when were they diagnosed, those are all things that you want to hone in on with your population because someone might have had, well, you actually had that for like a year, the doctor told you, and I know these discussions go on and diseases like ALS. So if that's important to the effect of the drug, it's important to make sure you characterize that as carefully as possible. With slow vital capacity, at least 50% might be an indicator there that tells you where they are in progression, but that would just have to be thought through. Interventions at 100 or 200 based on for the duration of the study, which is do they get there for 6 months? Stay there for 6 months. Okay. I didn't see anything about how you decided 100 or 200. And again, I just had a concept synopsis.
Yeah, those are kind of median doses that are already used in oncology.
So you can start with one and then go to the next
The
dose.
idea would be to do them simultaneously as kind of built-in dose finding.
Okay, all right. So then that leads to other and it's double blind. So that all has to be figured out how that drug gets labeled, how it's blinded, how it's distributed, who gets what. That has to be tied into some kind of WRS in order to do all of that. And again, where's the drug coming from? Who's doing the placebo matching? wasn't clear to me how frequently the assessments would be. And that would all help you in your rest of the day. You get 10 all done now. And then of course, the safety management plan, training, and again, day of election, training. So I'll end with everyone should do a clinical trial more than once, and have some fun.
I got a question.
Oh,
Could you share the slide at least with the 10 steps?
yes, yeah.
I mean, I took a picture, but I'm not.
Okay.
Anybody with other questions? I'm sorry I had to step out for department or whatever. You don't want to
Yeah,
go.
so.
Yeah. Okay.
Great. My email's up there, so anybody can email me anytime about how you're doing.
Dixie's an incredibly valuable resource, and now you know her, and you have her email, so